My name is Andrew Grant, and I'm an environmental science student in the iCons program at UMass. My background is in aquatic ecology, and I am here at the West Brook in Whately, Massachusetts, home of the study site for my honors research project. Since 1999, a team of researchers from UMass, USGS, and U.S. Forest Service have collaborated on a project studying the effects of streamflow and temperature on a population of brook trout here in the West Brook system. Among other things, they found that higher spring flows and cooler temperatures resulted in higher fish growth rates during those years. They also collected macroinvertebrate samples from the stream each year with the hopes of eventually linking the patterns in macroinvertebrate communities to the patterns in fish growth and population dynamics. My project is studying the effects of streamflow and temperature on the populations of macroinvertebrates in this stream. Macroinvertebrates are insects and other organisms that spend all or part of their lives living in the water.
They lay their eggs in the water and spend their juvenile or nymphal life stages under rocks and in vegetation until they mature as adults and emerge to mate in the nearby terrestrial environment. They include organisms such as mayflies or Ephemeroptera, stoneflies or Plecoptera, caddisflies or Trichoptera, true flies or Diptera, and Odonata, which are dragonflies and damselflies. These invertebrates serve as a primary food source for many fishes and aquatic food webs, as well as food for terrestrial animals, such as birds, spiders, and even bats. The most important groups of sensitive taxa are the EPT taxa, or Ephemeroptera, Plecoptera, and Trichoptera. These groups prefer fast-flowing, cold, well-oxygenated riffle habitats, whereas other groups such as midges, dragonflies, and damselflies prefer slower, warmer, and less oxygenated pool habitats and shallow, weedy ponds.
Macroinvertebrates can be used as indicators of water quality and stream health and can also be grouped into functional feeding groups based on their primary source of food. Functional feeding groups include things like scrapers and grazers, which eat algae off of rocks, filter feeders, collector-gatherers, and predators. These groups allow scientists to compare different sites from different regions based on their food web structure.
My project is investigating the relationship between streamflow and temperature on macroinvertebrate communities at the West Brook study site. The samples were taken in late June from riffle habitats and replicated 20-meter-long sections approximately 300 to 400 meters apart from one another. I am sorting and identifying the bugs down to the genus level and will be analyzing the samples based on the abundances of thermally sensitive and flow-sensitive taxa. For each sample, I will be calculating species richness and diversity. I will also calculate total biomass and total densities, as well as the densities of sensitive taxa, such as the EPT taxa. I will be comparing samples from years of high flows and low flows, as well as years of high temperatures and low temperatures. I am predicting that higher temperatures and higher flows will result in earlier maturation and higher biomass, whereas lower flows and higher temperatures will result in earlier maturation but lower biomass.
This project is interesting because it presents a unique opportunity to dig into a long-term data set and potentially shed light on the long-term dynamics of aquatic ecosystems. The main problem, however, is that I am barely scratching the surface of a very complex system where there are many other abiotic and biotic factors that could be influencing my results.
Climate change is expected to result in increasing temperatures as well as increasing severity of flooding and drought, with the Northeast predicted to become much warmer and wetter over the course of the next century. This past year, New England experienced one of the worst droughts in its history. Higher variability and intensity of floods and droughts will put additional selective pressures on aquatic organisms and will likely result in higher rates of extirpations of local populations of sensitive aquatic organisms and higher abundances of tolerant organisms. In order to preserve and protect these amazing creatures, it is important for us to invest in habitat restoration measures such as improving water quality and habitat connectivity in order to allow fish and invertebrates to have more suitable refuges to survive in during periods of stressful environmental conditions.